Hi students. Antibodies or immunoglobulins are glycoproteins produced by B effector cells or plasma cells. As per the clonal selection theory, upon exposure to its invading antigen, the relevant B cell gets activated and transforms into a plasma cell. Thereafter, it continuously produces billions of antibodies, which are nothing other than the secreted form of their own B cell receptors. Antibodies are named so as these are the proteins produced in response to incoming antigens. The term immunoglobulin rightly indicates that they play an important role in immunity and are gamma globulin by virtue of their structure. These are among the most abundant proteins found in blood. Glycosylation is their post-translational modification which has a stabilizing effect on them. Simplest immunoglobulins are Y-shaped molecules with two identical antigen binding sites or ABS, hence their valence is 2 per monomeric antibody. An immunoglobulin is a heterodimer made up of 4 polypeptides, of which 2 are heavy and 2 light. The two identical light chains consist of 220 amino acids each and have a molecular weight of 25 kilodaltons, whereas the heavy chains are of 440 amino acids and 50 kilodaltons. All of these are held together by interstrand disulfide bridges. The two light chains may be of either kappa or lambda type, but both always of a single type, and they never undergo class switching. On the other hand, heavy chains have 5 choices, namely gamma, alpha, mu, delta and epsilon, standing for IgG, A, M, D and E respectively. IgM isotype is usually first to be produced and later on a class switch is witnessed to IgG and many other classes. However, in a single antibody, both heavy chains are always alike at all times. Normally, IgG is approximately 80% of total antibodies in the blood, while IgA is found in body secretions. IgM is usually pentameric with a joining chain, IgA dimeric with a secretory component, and other 3 are monomeric. Based on minor structural variations in the heavy chains, IgG and IgA have subtypes too. Many more differences among these 5 isotypes are shown here on your screen. Amino acid sequence of both light and heavy chains varies tremendously at their N-terminal ends, but they remain constant at their C-termini. Light chains have 2 immunoglobulin folds of or domains of 110 amino acids each. Its upstream is variable VL and subsequent downstream one is constant CL. Likewise, a single upstream fold of a heavy chain is VH, followed by 3 or 4 constant domains called CH1, 2, 3 and 4. In addition, IgG, IgA and IgD have a hinge region shown here that provides great flexibility to the molecule so as to access the epitope of the incoming antigen more comfortably. IgM and IgE are observed to lack the hinge region. Now, one VH and one VC together form one ABS or antigen binding site, and there are 2 such ABS per monomeric antibody. The variable region VH of the heavy chains is also about 110 amino acids long, but the heavy chain constant region CH is about 3 to 4 times longer, which is 330 or 440 amino acids, depending on its class. It is the N-terminal ends of the one light and one heavy chain together which constitutes a single antigen binding site or ABS. The maximum amino acid diversity in the variable regions of both light and heavy chains is confined to 3 small regions in each chain called complementarity determining regions CDRs or hypervariable regions. The remaining parts of the variable region known as the framework regions are relatively lesser variable. Starting from the amino terminal, These regions are numbered CDR1, 2 and 3. The CDR3 is most variable of the three CDRs. It is these regions which provide the specific binding and complementarity of an antibody with its antigen. Somatic hypermutations happening at these CDRs at all times also contribute to so-called affinity maturation, a special property of antibodies that improves its affinity to its antigen tremendously over time after the first encounter of antigen and antibody. We shall be discussing these amazing and unique concepts in more detail and deep in our future videos. We hope, students, that this oversimplified account is empowering you to easily grasp and enjoy immunology. Forthcoming videos in the antibody series would include the understanding of the topics shown on your screen. We believe that imparting knowledge in simple language and small doses accompanied by vivid visuals works wonders. You are welcome to provide us your viewpoint on the same. Till we meet next, stay blessed, keep studying, and bye for now.